think we should
Thank
move
you.
on to our next presenter, who is Pepper Landson from Praetego.
Thank you so much, Sonia. I appreciate it. Let me go ahead and share my screen. Let me go to that and we do share. And are you seeing my one giant kind of watery blue-green
Yes.
slide? Okay, fantastic, fantastic. Well, thank you so much for the opportunity. Metabesity this year has been profound. And as always, I learned so much, and I just am so amazed by the work of so many people around the world to further this cause. Praetego is Latin. It means to protect. And I'm Pepper Landson, the co-founder and CEO. Praetego is mission-driven to limit disease progression in the chronic diseases of aging. And throughout the many wonderful sessions I've seen this week at Metabesity, the focus seems to have been on the biology of aging, what it is, what it might be, and how we might address it as a community. To help frame up your appreciation of Praetego's technology, we're focused on addressing the chemistry of aging. Praetego offers a first-in-class technology able to inhibit advanced glycation end products at the step of formation. The AGE pathway is implicated in serious diabetic complications and the chronic diseases of aging. We have extensive development data in a variety of indications, and we've selected our lead candidate. AGE pathology is not specific to a single disease. As such, our technology lends itself to a broad spectrum of indications. Our first target is diabetic microvascular complications, specifically neuropathy. With no drug approved to limit disease progression, our lead program presents an $8 billion market opportunity. This technology was developed in-house, has robust IP, and a significant market potential. Praetego's team has over 100 years of drug development experience, and we've been studying the AGE pathway and AGE inhibitors for over 30 years. We're now raising funds to move from a preclinical stage through early clinical proof. And with your help, Praetego will harness the power of protection. Considerable evidence links AGE formation to the pathology of diabetic complications and other conditions associated with oxidative stress. AGE formation occurs normally due to the essential presence of glucose in metabolism. For example, AGE levels in skin collagen along lived tissue steadily increase with age, and there's a lot of evidence that points to AGEs as the cause of wrinkles. Skin AGEs can be measured by autofluorescence, and AGEs are formed by indiscriminate glucose reactions with proteins and lipids, glycoxidation and lipoxidation. These reactions produce the antagonistic byproduct of an advanced glycation end product. AGEs then generate ROS and other undesirable downstream consequences. Biochemical defense mechanisms normally protect us from the deleterious effects of AGE formation, but this process gets stressed with the onset of hyperglycemia and diabetes and acute stressors like trauma. Whether the insult is acute or chronic, the AGE pathway and resultant oxidative stress drives pathology. Now, given the sophistication of this audience, I offer this slide as a reminder, not of the many conditions and diseases linked to oxidative stress, but of how easily we get distracted by excessive information. At Praetego, we're not aiming to be all things to all people. Rather, we're focused on the areas where the science, ours and that of the science and peer-reviewed literature, support our AGE inhibitor candidates, specifically diabetic complications and neurodegeneration. To put solid points up on the Gero Science board means two things: a win for patients and more investment into this field. And these were recurrent themes I heard this week at Metabesity. Praetego's candidates are called Amadorins, and they inhibit AGE formation. Specifically, the Amadorins are chemical modulators of the redox metal ion catalysts of oxidation, and they do this through a bidentate mode of binding. Amadorins are not chelators. Rather, they diminish the reactivity of the redox metal ions through transient binding. By interfering at this initial step in the pathway, formation of AGEs is interrupted and the burden of AGEs is reduced. For diabetics, this means protecting end organs. For aging, it could mean a slowing of one of the known drivers. In summary, AGE formation is a chemical driver of diffuse pathology. Glucose is the fuel for the AGE pathway. Our bodies deploy antioxidant defenses to protect us from AGE-driven pathology. If we're diabetic or experience trauma, these defenses can be overwhelmed. Reducing the burden of AGEs offers the potential to slow or prevent disease progression across the spectrum of age-related diseases. Amadorins, as potent AGE inhibitors, are unlike traditional pharmacotherapy. They circulate in plasma and function as chemical protectants. They act upstream of a pathogenic process. Hence, they are protective and possibly preventive rather than interventional. Starting with our two lead candidates, Praetego is pursuing diabetic microvascular complications and neurodegeneration. With sufficient funding, we will be in clinic late next year. Benefiting from a lengthy history of studying AGE inhibitors, Praetego was founded in 2017. To date, our funding is thanks to founders, angels, and the NIH. Specifically, this technology has been validated by 3 NIH awards so far, 2 from the NIDDK and the latest from NIA. We continue to cultivate our NIH relationships to further our novel candidates. The challenge with this type of money is the time cost. Hence, a focus on raising a Series A to accelerate our progress to and through clinical trials. Our mission is to get a suitable candidate to clinic as soon as possible. Important missions require remarkable teams. And Praetego has the benefit of a robust talent pool in the RTP area of North Carolina. As the co-founder and CEO, I deploy 3 decades of know-how in operations and drug development. I'm a proven value creator in early stage ventures. My contributions to infectious disease, oncology, and neurology drugs have created many first-in-class commercial successes. Raja brings the foundational science to our story as the inventor of the Amadorins. A world-class chemist, he has led translational science in 3 companies. Katina is a life sciences exec with a strong track record in business development and fundraising. She draws from 25 years of advising corporate clients on capital markets, fundraising, and strategic transactions. Joanne is a seasoned executive with extensive expertise in defining product development strategies and executing on those strategies. As diabetic complications span multiple specialties while sharing an underlying pathology, we've curated a cross-sectional team of experts. They are accomplished clinicians and researchers. They provide key intel on what's desired in clinical care and how best to design trials to satisfy regulatory authorities. As the Metabesity conference demonstrates, gathering the brightest minds from the spectrum of stakeholders is how we drive equitable progress. So with a stellar team, we answer the call to develop an AGE inhibitor candidate. We know AGEs are a druggable target because 3 companies conducted clinical programs using different mechanisms, and 2 made it into pivotal trials. The first was Alzheimer with a drug called aminoguanidine. It was an effective scavenger of the downstream chemistries in the AGE pathway. They did make it through a pivotal program in diabetic kidney disease, but toxicity killed that candidate. Nephrogenics took the first Amadorin into phase 3, also for diabetic kidney disease. Unlike aminoguanidine, that candidate inhibits AGEs at the first step of formation. It also had an exceptional safety profile. And the drug did demonstrate clinical efficacy, but unfortunately, Nephrogenics was unable to complete the pivotal program. So that leaves 2 preclinical programs targeting AGEs, Praetego and Rebel Therapeutics. Praetego did a meta-analysis of the clinical data from the Nephrogenics diabetic kidney disease program. It demonstrated the renal protective effect of that first Amadorin. Specifically, those on placebo were twice as likely to progress to end-stage renal disease within a year than the patients on active drug. And this matters because AGE pathology is a known driver of kidney disease. This data offers related clinical proof for the Amadorin promise. With a way to measure AGE inhibition, that first Amadorin became our benchmark. Studying the structure and function of that first candidate, we created many novel candidates. Using our AGE assay, we refined each generation for potency. Our lead candidates are 2 orders of magnitude more potent than that original prototype. And so, having made what's likely to be the maximum increases in potency, we're now focused on structural modifications to maximize safety and bioavailability. By improving potency, we saw better performance in vivo. The graph on the right reflects improvements across those generations in nerve conduction velocity in an animal model of early diabetic peripheral neuropathy, or DPN. Data from the lead candidate appears in just a moment. Now, our candidates are the product of decades of research with a remarkable amount of data to demonstrate merit and proof of mechanism. Determined to develop the ideal AGE inhibitor, we've assembled the key components of a viable drug: safety, potency, and patent protection. The common thread amongst these 4 generations is the amelioration of diabetic peripheral neuropathy, which leads me to our lead candidate, PTG6. Through our preclinical work to date, we've determined that it offers a wide margin of safety, an extremely low risk of drug-drug interaction, and we know it reaches the tissue of interest. These are all desirable features of a chronic use drug. As a possible first indication, like all previously tested Amadorins, PTG6 demonstrated the ability to ameliorate nerve conduction velocity in an animal model of DPN. Now, recalling its potency relative to the earlier generations, it performed at a very low dose, which bodes well for a wider therapeutic window. Now, as anybody who's worked in DPN knows, affecting NCV in an animal model has never translated to clinical efficacy, and many would argue that nerve conduction velocity is not the appropriate endpoint for clinical trials. So perhaps this data will go farther to demonstrate the promise of PTG6 as a disease-modifying candidate. Corneal confocal microscopy measures Bowman's fibers in the cornea, and the density of these fibers is an indicator of peripheral nerve health. Using CCM, we saw that PTG6 restores small nerve fibers in our animal studies. And the FDA is considering small nerve fiber measurements such as CCM as the future of DPN clinical trial endpoints. The development plan for PTG6 considers the broad application from its mechanism of action. Specifically, we're accommodating the current regulatory pathway of a single indication IND, such as DPN, for our first step into clinical programming. We'll conduct early clinical trials to include safety data in healthy patient volunteers, followed by type 1 and type 2 diabetics. And then our phase 2A is designed as a target engagement study. It will have biomarkers and surrogate endpoints of the diabetic microvascular complications, namely neuropathy, retinopathy, and nephropathy. A successful phase 2A will allow us to springboard into multiple indications and accelerate development timelines. Laying out the de-risking events and the cost to each milestone, our plan is this. The first $5 million gets us into clinic in about a year. The next 11 million over 24 months gets us early clinical proof of concept. We're already speaking to potential strategic partners who await clinical data before partnering. As designed, we deliver substantive clinical data in 36 months for 16 million. Our primary exit plan is a strategic deal upon clinical proof. The trials for phase 3 are extremely expensive and large, and therefore a strategic partner makes the most sense. But if that is not the route we take, we could raise Series B and continue development ourselves. Thank you so much to Connexum and Catalyst for creating an amazing showcase this year through the Metabesity conference. This community is tasked with the stewardship of science to improve health span, and Praetego is honored to be a part of that. Thank you.
Thanks, Pepper. That was great. Lots of really great data. I like that as an investor. I like that you have a lot of experience. I'm always happy to see a founder who's been through a number of companies. And then my next question is: so why this company? Why? What is it about this company that you're passionate about, and how do you think it's going to change the world?
Sure. So this company is essentially the final steward of the technology, which is the Amadorins that I worked on. I used to lead the program for nephrogenics, so I was there when it all fell apart. I believe in the science, and I believe in what this science could mean both to an aging generation as well as the generation behind me. And it's in an area, obviously, metabolic diseases that has been underserved by the investment community for a very long time. So as a self-proclaimed champion of the underdog, I think the science is right. It's not the shiny new science. This is old school pharma development. It's a small molecule, but it works differently. So that is the sexy bit. It's not one of the typical small molecules that binds to a cell and does something great while doing something questionable. Instead, as a chemical protectant technology, we can actually work as adjuvant therapy in addition to the other things like some of the new class of diabetes drugs that are doing really wonderful things. We don't need to compete. We can be collaborative and cooperative and benefit everybody who winds up on our drugs.
And could the Amadorins be utilized for other diseases as well, aside from diabetes?
Absolutely. AGE pathology is published to be a factor in all the chronic diseases of aging. We just want to focus on what we know we can move the needle on, but I have grand plans around, let's get a win on the board in a diabetic microvascular complication like neuropathy. I just got data literally like 20 minutes ago that we know where our drug gets into the retina. So retinopathy is probably our second indication, which would be fantastic because that's an indication that's ripe for intervention and definitely disruption because the current options are pretty lousy. We know we've got an active study with our lead candidate against neurodegeneration, not a diabetic model. We'll have data on that late this year. So beyond neurodegeneration, you've got potential in arthritis, right? Again, a chronic inflammation-driven disease and a disease of aging. And well beyond that, I'm just trying to practice discipline around, let's focus on what we know we can affect change on and the science supports it, get that win, build credibility with the FDA, and then try to break through some of these silos within the divisions of particular indications.
Right. It's the whole body's aging.
Exactly, it's a closed system.
Yes, right, and there's an impact across all things. So, well, that's great. I think we're running a little bit behind time, so I think we're gonna go to the next presenter now. Thanks, Pepper.
My pleasure. Thank you.
Thank you.